Right, so we're going to take a look at the actual ways in which antibodies function and start to talk about where they come from. So antibodies function in several different ways. There's three ways in particular that you need to think about them, and these all overlap, particularly these last two to a certain extent. You can think about antibodies as having neutralizing properties. That's perhaps the simplest way to think about it. But they also have two other important properties which are referred to as opsonization and complement activation. I'll just go through briefly with you what those roles actually, what those words actually mean. Neutralization is kind of the obvious one. So antibodies can bind very specifically to certain structures. Now these might be very small structures like toxins, so small toxic proteins. They could be larger structures like viruses or bacteria that they recognize. The idea here, though, is that all the antibodies do is they block the activity by binding to the active parts of those structures. So here's an example. So these first three diagrams show an example of a particular kind of toxin. So this could be, for example, an anthrax toxin or a tetanus toxin, something like that. These toxins often come, incidentally, in multiple parts. So often there are two or three peptide chains associated in a toxin molecule. And what you can see here is an example of a toxin. This toxin has to recognize the cell that it has an effect on. It does that by binding to particular receptors on the cell. Having done that, it's then endocytosed, so it's taken up into a lysosome structure, a phagosome or an endosome structure like that. And then the active part of the toxin, in this case this red dot here, is pushed out into the cytosol, and that's where it exerts its toxic effect. So there was a seminar about this in the context of anthrax toxin about a week ago, that some of you, I think, came along to. So if you've got an antibody against that toxin, all that's happening is the antibody is simply recognizing the toxin and binding to it. And if this is the part of the toxin here that recognizes the receptor, and it's got an antibody molecule sitting on that, it can no longer recognize the receptor. So all the antibody is doing is covering up the part of the toxin, in this case, that's needed for the toxin to bind to the cell. As long as that part is covered up, the toxin can no longer act. And this is the basis, incidentally, for some anti-venoms, some treatments that, if you ever have the misfortune to be bitten by a snake that's a venomous snake, try and get yourself along to a doctor that's got a suitable anti-venom as long as possible. The anti-venoms often just bind to the toxins within the snake venom and stop them from interacting with your cells. Clearly, the quicker you can get that done, the better. Here's another example. That was an example of a small molecule, a toxin. In this case, here's a virus. Same basic kind of process takes place when a virus infects a cell. First of all, it's got to recognize that cell. So there's got to be an interaction between some structure on the viral cell surface and some receptor on the cell surface. That virus may then be engulfed by the cell and then undergoes a structural change so that the nucleic acid from the virus is released into the cytoplasm, where it can be transcribed or translated and turned into more viral particles. But again, if the antibody recognizes structures on the virus which are required to recognize the receptors, that's going to block the activity of the virus. And the thing to notice with these sorts of events is that it's not getting rid of the virus is still present, but it's no longer able to act. Same thing again, this is antibody neutralizing bacteria. So bacteria as pathogens act in all sorts of different ways, of course. Not all of them go inside cells, but some of them do, like salmonella, for example, gets engulfed into cells. And typically that will happen again via some kind of receptor recognition. Bacteria produce proteins like adhesins that enable them to stick to cell surfaces, and they then become internalized and can cause infections. But again, antibodies can bind those adhesins and block cellular uptake. So again, the bacterium isn't removed from the body, but it's just prevented or reduced in its ability to cause infection within the body. Opsonization and activation of complement are different to this. So neutralization is the simplest way in which antibodies work, but they don't remove the offending item, if you like, opsonization and activation do that. So they help the body to destroy the invading pathogen. Opsonization is essentially where the entire structure becomes coated with antibodies, and as we'll see, that then not only neutralizes it, but also makes it recognizable by some of the cells in the body. And complement. Complement is not just one protein. Complement's a whole set of proteins with a range of antibacterial activities. And complement, I'm not going to go through all of these, it's not that relevant for this particular course, but complement can act directly by binding to cells, but also by binding to antibodies that are bound to cells. So that's another way in which cells can be recognized by toxins or bacteria or invading items can be recognized by cells of our immune system by the binding of complement. Why is this useful? It's useful because certain cells in the body contain receptors that recognize antibodies, and they particularly recognize the Fc part of the chain. Remember, just to go back to that diagram, this one here, the Fc part is the tail of the antibody. This is the head up here. This is the tail of the antibody. So this is the part that's recognized by particular receptors in the body called Fc receptors. And complement protein is also recognized by receptors called, not surprisingly, complement receptors. So what the binding of antibodies does in this case is it makes the foreign item, the toxin, the virus, the bacteria, cell, whatever, more recognizable by cells of the immune system than would otherwise be the case. So here's a diagram of how this can happen. So we've got an invading pathogen of some sort here. Obviously, this is not to scale. Some protein on the cell surface of the pathogen has been recognized by the antibody. The antibody Fc component is then being recognized by an Fc receptor. And a cell that expresses Fc receptors called an effector cell. There's loads of different cells which are effector cells. And that can then lead to phagocytosis of the antibody bound to the structure. So the whole lot goes inside the cell. Typically, these cells will be things like macrophages or neutrophils. Again, cells that have the property of engulfing foreign structures and then breaking them down. But in order to do that, they've got to be expressing the right receptors, the Fc receptors to recognize the antibody and the complement receptors to recognize the complement protein. So here's another diagram from Janeway's that illustrates this rather nicely. This is showing both effects, basically. So you've got the antibodies here. These are the structures shown in yellow. You've also got complement, which is the kind of bluey structure here. As I said, there's lots of different kinds of complement. This is a particular kind of complement called C3b. The antibodies are exposing their Fc regions, which are then bound, then bound by these Fc receptors, which are the little green sticks here, and the complement is bound by the CR1 receptor. This is a macrophage, so once the organism which has got those antibodies and the complement stuck to it is bound, it becomes endocytosed, produces the phagosome, and that then fuses with the lysosomes, as we've discussed already, and the invading bacterium is broken down. So the advantage of this sort of process is that you actually remove the toxin or the virus or the bacterium from the body, so there's no chance of it causing reinfection anywhere else. The other thing that happens, which is important, is that complement not only helps cells to be recognized through the complement receptor, but it also directly punches holes in cell membranes and produces something called complement-mediated cell killing. So the binding of complement is both a cytotoxic effect in its own right, particularly with bacterial cells, but it also helps those cells to be recognized by the macrophages and other cells that can gobble up the antibody and complement-coated material. So this diagram tries to bring all these different things together. So if we go back to the first one, we've got toxins. Here's some simplified version. Toxins binding to cell receptors, antibodies binding to those toxins, preventing them from being acted on by for them acting on the cell-based receptors. Eventually, of course, these structures themselves will be recognized by the FC receptors on the macrophages and broken down. We've got opsonization, so coating of the bacterium, in this case, with antibody, and then it being bound by FC receptors and broken down. And we've got complement binding directly to antibodies which are bound to the bacterial cell. And what it shows here, this little thing here, is showing how complement can actually punch holes in the cell wall of the bacteria, and that causes the bacteria to lyse because the internal material in the bacterial cell spills out. Now, you can appreciate all these things overlap to a certain extent. Complement's got a dual function. Neutralization will eventually be followed by uptake through the FC receptors. So these are all, to a certain extent, different facets of the same process. What's important to get from this is that the role of antibodies is to act in several different ways. So they're not just binding to and neutralizing stuff, they're actually actively promoting lysis in the case of bacteria and actively promoting uptake of the invading organisms or invading material into cells that can deal with them. Obviously, that works fine for things that are small, so viruses and bacteria are much, much smaller than human cells. There may be situations where you actually want to destroy things which are too big to be taken up by a macrophage. So, for example, the body, if it hasn't succeeded in dealing with the virus, may want to recognize and destroy a cell that's become infected by a virus. And in that case, phagocytosis isn't going to work because the macrophage isn't big enough to be able to take up another human cell. This is where cells like the natural killer cells come in, and these also carry FC receptors. So here's an example of what can happen here. So here we've got a target cell. Maybe this is a cell that's become infected by a virus, so it's expressing some kind of viral protein on its cell surface. The antibodies are recognizing this and binding to it. So this cell is now coated with antibodies. Again, these FC regions will be sticking out into the surrounding fluid, and NK cells, natural killer cells, contain FC receptors that when they bind to these become activated and lead to the secretion of a whole range of different things that will destroy the target cell. So this doesn't involve one cell being taken up by another cell, it's just a way of attracting killer cells into the vicinity of the cell that they want to destroy. This is known as ADCC, antibody-dependent cell-mediated cytotoxicity. So it's antibody-dependent because it requires the antibodies to bind to the target cell. It's cell-dependent because it requires a natural killer cell to arrive and destroy the cell that the antibodies are bound to. And cytotoxic simply means that it's cell-killing. And this is actually quite an important process, as we'll see shortly, in some kinds of cancer therapy. Now, the natural killer cells themselves have got a whole range of different things that they can unleash. They can produce these proteins called perforins, which go into cell membranes and make holes in them. And they can also then produce proteases, which can pass into those cells, and the proteases can trigger apoptosis. And apoptosis, most of you probably know about this, is a process of programmed cell suicide. And it's different from cell lysis. So in cell lysis, the cell simply breaks open and spills its contents into the medium. In apoptosis, the cell essentially breaks down all its internal compartments without breaking open. It will eventually lyse at the end of the whole process, but by then everything inside the cell will have been reduced to fairly small molecules. And the advantage of this is pretty obvious. If you've got a cell that's infected by viruses, you don't want to cause that cell to lyse, because all that will happen if you do that is you'll spill lots and lots of viruses out into the surrounding material. But if you can cause that cell to undergo suicide and destroy all its internal contents, including the viruses, then you're going to get much less problems with release of material if you go through that route than if you go through the direct lysis route. Now, people have exploited this. So, for example, there's a kind of various kinds of cancer therapies that involve antibodies against cancer cells. An example is Rituxan. And Rituxan is a monoclonal antibody that recognizes particular proteins which are expressed on the surface of cancer cells. And the reason this is very useful is what happens is the rituxan will bind to these receptors. Because it's an antibody, it's got an FC region. That FC region is recognized by natural killer cells. So what will then happen is that the natural killer cells are attracted to the cancer cells and then kill them off. So it's a way of essentially engineering antibodies to enable the antibodies to turn a component of the host's immune system against the cancer cells, against the cells that we want the therapy to destroy. Okay, so hopefully from all of this you're getting some idea of how antibodies work, why they're important, beginning to get a sense of how they can be used therapeutically, and how in order to understand their therapeutic action, you need to understand quite a lot about how they function, and as you'll see later on, about their structure. So what we're going to look at now is the process of development of the adaptive immune response. It answers the question of where do antibodies actually come from, what are the steps that take place between an infection taking place and the cell having a fully fledged antibody response against that infection. As we said, this can take some time. We may be talking several days here. And most infections that get past the innate immune system and stimulate an infection like this are what we'd refer to as self-limiting. They're not particularly pleasant when we've got them. We'll be stimulating an inflammatory response, one of the consequences of which is a fever, which is actually quite a good thing to have as long as it doesn't get too high. But after a few days, your body's immune system will learn to recognize the infecting organism or pathogen, and it will be able to clear it using some of the mechanisms we've talked about, plus the T-cell mediated mechanisms that I'm not going to discuss. So a very large number of infections we never even notice that we've got because the immune system deals with them. And then occasionally we'll get something that will knock us out for a short period of time, but eventually we'll cope with it. Even something like a nasty flu bug, eventually the immune system will recognize that virus and be able to clear the infected cells and the infecting viruses from the body. Obviously, the pathogens are evolving to get around that. That's why flu is so nasty, because it's constantly changing the appearance of its cell surface. But a lot of illnesses that we would get, we don't even recognize. A lot of illnesses that we do get, we do recognize. We just feel crap for a few days with a high fever and generally no energy. That basically means our immune system is looking after us, and within a few days, we'll be sorted. We particularly get these in childhood, at least we get them if we're not vaccinated against them. So in the days when people weren't vaccinated against the standard childhood diseases, so people like me that didn't have all those vaccines, I had mumps, I had measles, German measles, chicken pox, all viral infections, none of them terribly nice, but they never last more than a week or two. Having got them, you're pretty much then immune for the rest of your life. Most of you will have had the fortune of being vaccinated against most of those, although some of you may still have had things like chicken pox. That's a classic example of something that your immune system recognizes pretty quickly and deals with. Of course, a lot of these diseases can have unpleasant side effects. So German measles is really bad if you're pregnant. Measles can cause complications that lead to brain damage. So even though they're often referred to generically as childhood diseases, they're still not particularly nice ones to have around. Okay, what's the process that takes place? So you can look at the immune response, the adaptive immune response, as being broken down in these different stages. Initially, of course, we've got infection. Some kind of infecting organism has got past all the barriers, the physical barriers and the innate immune system barriers, has got into the body and is beginning to grow and replicate. So we're starting to have a foreign population of organisms living inside us. At some point, the immune system spots this. The amount of a particular antigen or set of antigens expressed by that invading organism gets to a point where it turns on the adaptive immune system. And then very quickly you'll get the induction of the adaptive response. And this may be over the space of a few days. Once that adaptive immune response has kicked in, it will then, hopefully, if it's working okay, and if the pathogen hasn't got the upper hand, it will clear the pathogen from the body. So it will attack and destroy the infected cells, it will attack and destroy the pathogen. So eventually there'll be none of that pathogen left and we'll feel better, which is nice. The fourth stage is immunological memory. That process having happened, if you get reinfected by the pathogen, the immune system will respond much more quickly next time around. It's learned to recognize that particular pathogen or some component of what that pathogen expresses. So the question is, where do antibodies come from? There's lots of different answers to this that we'll go through, but I'm going to talk about this idea of clonal selection. So just to remind you, this is the family tree of antibodies. They all start off as stem cells, obviously. The antibody-producing cells are the B cells. What you eventually finish up with, once you've had an adaptive immune response, are two kinds of cells, both of which I'll explain. Plasma cells, which are the ones which are churning out the antibody, and memory cells, and these are the ones that will trigger the adaptive response if we have a second infection. Now, the idea about how this might happen, about how we have all these antibodies recognizing all these different kinds of antigens in the body, is we start off with the progenitor cell. So this is initially this stem cell here, then the partially differentiated stem cell that's going to give rise to all the different cells of the adaptive immune system. And this cell differentiates to give rise to lots of different early lymphocytes that produce different kinds of antibody. Now, there's a real danger here, which is that lots of these things that they recognize might be self. And what you really, really don't want is your immune system to start recognizing yourself as being foreign. So a process takes place very early on in development of the immune system. This is mostly shortly after birth, where any cell that's producing an antibody or has got a receptor on it that recognizes a component of the self is destroyed. The cell takes these self-recognizing cells out and basically makes sure that they're not allowed to proliferate. So what you've then got is a pool of what are called mature naive lymphocytes. So what that means is that these are lymphocytes that contain receptors that don't recognize self, so they don't recognize any component of ourselves, and they're ready to recognize foreign antigens. So when an infection comes along that presents a foreign antigen, the one that recognizes that antigen then proliferates. So you then get many, many copies of that cell, all of which can recognize that same antigen. So you've now got a whole group of cells that are capable of producing the antibody that can remove and deal with that antigen. So we've essentially got two steps of selection taking place here. One that removes all the proteins, all the cells initially producing proteins that recognize self, and then another one where the ones that are eventually stimulated because of an infection massively proliferate and produce large amounts of antibody. This is the clonal selection hypothesis, and what we'll see is this is mostly true, but there's actually quite a bit more to it than this. One of the questions that you might be asking, and we'll pick this up later on, is where does this diversity come from in the first place? Where does the ability to produce lots of cells with loads and loads and loads of different recognition capabilities actually come from? There isn't enough space in the human genome to encode all these different antibody types. So this was a big mystery for many years. We now understand it very well, and we'll go into that later on. Okay, so that's just to summarize what I've just talked about. Okay, so we have these two cell types, B cells and T cells. We're going to focus on B cells, but as you'll see, T cells come into the story as well. Now, B cells express receptor proteins, and they express a particular receptor protein which is fairly unique to that individual B cell. And these are called B cell receptors or B cell receptor complexes. You can see a diagram of an example here, and what you notice straight away is this looks actually quite like an antibody. You've got a heavy chain here, you've got a light chain here. This heavy chain is a little bit bigger than the standard heavy chain from an IgG, but it looks very, very much like an antibody. And that's not surprising, because actually it's very closely related to a standard antibody. So these are all evolved from the same starting point. There's some other components here which are very important. And the way that these work is that if this receptor molecule recognizes something, conformational changes take place that lead to the transmission of a signal to the inside of the cell saying, we've found something, we've spotted something, we now need to start doing the things that B cells do. Now, you'd think if you designed this system from scratch, that signal would be to proliferate, to make lots of copies. But it turns out it's a good deal more complex than that. What actually happens is this, and I'm going to go through this fairly quickly, because I want to get on to talking about structure in the next lecture. If you find it a little bit complicated, then do go away and read up on it in a bit more detail. So the first thing that happens basically is the B cell receptor recognizes a foreign antigen. That might be, again, a toxin, it might be a protein on the outside of a bacterial cell, it might be a viral protein, all sorts of different things that it could be. That causes a signal to go to the inside of the cell to say that something's been recognized. What the B cell then does is it takes in that whole receptor complex with the antigen bound to it, and it scrunches it up. It basically cuts it up into small peptides. Having done that, it takes the peptides from the antigen and it puts them back onto the outside of the cell. And it puts them onto the outside of the cell bound to a very particular protein complex. And this protein complex is called the MHC class II complex. MHC stands for major histocompatibility, but you just need to know it as MHC class II. Now, I'll show you a diagram of this in a minute if this is a bit hard to follow. This peptide bound to this MHC class II, which is now on the outside of the B cell, is now recognized by a T cell. This is where the T cells, or some of the T cells, come into the story. And these are called T helper cells because they help the B cells induce this immune response. And this T helper cell also recognizes this antigen. So it will only recognize the antigen if it's bound by a B cell and presented to it on an MHC class II protein. There's also quite a lot of other structures involved in this process, so there's a lot of cells cozying up to each other and recognizing each other that has to happen. I'll show you a diagram of that in just a moment. When that recognition takes place, the T helper cell now produces cytokines that stimulates the B cell, the one that presented the antigen to the T cell, to now differentiate, make lots and lots of copies of itself into a plasma cell that's going to secrete antibodies. And the recognition by that antibody will be the same as the recognition by the original B cell receptor complex and by the T cell receptor complex. Okay, so here's one diagram. Here we've got a B cell. It's expressing a B cell receptor on its surface. That B cell receptor here is recognizing an antigen, some part of a virus in this case. That is stimulating the B cell to take that whole complex, the receptor and the bound antigen, into the cell. The peptides are then being put back on the outside of the B cell on this little yellow thing here. This is the MHC class II complex. And that's been recognized by a T helper cell that's also capable of recognizing that peptide. Now, the T cell can only recognize the peptide if it's bound to the MHC class II. It can't recognize it when it's bound on its own. There's other proteins as well that talk to each other between the T cell and the B cell. Once that whole recognition has taken place, the T cell is now releasing cytokines, which is stimulating the B cell to grow and differentiate. Once it does that, it produces antibodies that are based on that B cell receptor. Okay, so B cell recognizes something, presents it to the T cell recognizes it, stimulates the B cell then produces the antibody that recognizes that same thing. There's even more to it than that, but I'm going to leave the next stage to a later lecture. So here's a diagram again showing us in a bit more detail. You've got the B cell receptor here, BCR, binding to an antigen. This is the part of the receptor that stimulates the cell. The antigen and the receptor is taken inside, processed to short peptides, presented on the MHC class II peptide. That's recognized by the T cell receptor. There are these other proteins as well, the CD4 is very important, that recognizes the MHC class II. These things talk to each other. Once that recognition has taken place, cytokines, interleukins in fact, are being produced to stimulate that B cell to grow and produce more antibodies. So it's a pretty complicated process. And there are places where this can go wrong. One of the things that interferes with this drastically is infection with HIV that targets some of these proteins that the T cell has on its surface, which is why if you've got an HIV infection, your immune system doesn't function anymore because this whole process is broken down. Okay, so the cytokines that are produced by the T helper cell cause the B cell to proliferate, to make lots of copies of itself, and when it's made all those copies and fully differentiated, it's now going to secrete the antibody that's got exactly the, well, actually not exactly the same specificity as we'll see, but the same basic specificity as the B-cell receptor did to begin with. In fact, during that process of differentiation, the antibodies get better and better at recognizing the antigen, but we'll say more and more about that in a later lecture. Okay. So here again is the whole thing in a diagram from Genway. You've got the B cell recognizing the antigen, presenting the antigen as a small peptide on the MHC class II, the T helper cell stimulating proliferation of the B cell. B cell grows, divides, and ultimately produces two different sorts of cells. The plasma cell that basically churns out antibody, so that's engineered just to make lots and lots of antibody. The antibody recognizes the same antigen. And the memory cell, is similar to the original B cell, but is now better at recognizing the antigen than it was before. And again, we'll see how that happens a little bit later on. So there's two things which are happening here. One is the immune system is being stimulated to produce cells that make lots and lots of antibody. And the other is the immune system at the same time is getting better at recognizing the original antigen. Okay, so there's a real learning process that takes place. And this is why if you get a subsequent infection, the process is much quicker. So the T cell is, the immune system is now already pre-adapted to recognize that particular antigen. So here is a longer-term diagram now. This is in days and eventually in years about what's going on with the immune system. So the first infection stimulating an immune response, that's what we saw before. What will happen then is the antibody titer will go up and down again as the infection builds, but then as the immune system clears the infection from the body. That's nice because what it means is that if we get infected later on, although there'll be a jump in the antibody level, we actually won't know that we're ill. So the immune system will cope with, if you've been vaccinated or if you've had a disease like chickenpox or mumps, it's not that the virus has gone away. You're getting infected by it all the time, but the immune system just deals with it straight away. You don't even know that you've been infected. The only way that you can tell you've been infected is the levels of circulating antibodies will transiently go up, and then they'll go back down again once that thing has been cleared. But without looking at the antibodies that are circulating in your blood, you would have no